Thank you so much, everyone. It's an immense privilege and honour to be here. My name is Samuel McKee. I am a DNA repair researcher at the University of Reading in England, just outside Oxford. I'm also a tutor in history and philosophy of science at Manchester Metropolitan University, and I do some research with the University of Cambridge as well. Greetings from the Mars Society UK as well. I promised the guys back home that I would say that. I'm an ambassador for the Genetics Society and an associate with the Royal Society of Biology. Part of the challenge here of being English is that because of your accent, people assume that you're smarter than you are. But here we go. We'll just kick off with our best. My team, I'm part of a team at the University of Reading that is involved in bioinformatics and computational biology. So we, as you compete with AlphaFold in the various tournaments and competitions, my PhD project is in simulating DNA damage and repair protein responses, particularly linked with mismatch repair. So my focus is primarily on colorectal cancer and Lynch syndrome, but there is obviously this crossover with the space sector because space is really horrible to DNA. And in particular, it's the worst kind of damage that we're dealing with, complete double-strand breaks. So at the moment, I'm working on a project where we are structurally assessing the various proteins involved with double-strand break repair and cataloging their different variant positions, sites of interest, and seeing how perhaps when it comes to cancer, we could generate some new inhibitors, perhaps, that could address the problems, the cancerous problems that come when these break down. So at the moment, though, we're just going to talk about how we can keep astronauts alive a little bit better. Alive on the way to Mars, on the way back home from Mars, assuming they want to come back, and while they're there as well, because space offers little of the protection that Earth affords. This is all stuff I know you're very, very familiar with. I generally find that when it comes to DNA repair and space, there's three camps of people. There are those who say it's not a problem at all, nothing to worry about, nothing to see here, everything's going to be just fine. Then there are those who say that shielding is all we need to do. Basically, if we can generate a lot of shielding, then everyone will be fine. We'll just sort of encase them in a bubble the whole time that they're there and back. And if we can generate the right shielding, it'll be fine. And then there are those, like everyone at the University of Reading who I work with, who says this is crazy. There's no way anyone can survive 5 minutes on Mars. So it's an insurmountable problem. I do find that when it comes to radiation damage, the overwhelming majority of the conversation is based around shielding. And at the moment, that seems a little unnecessary because we're at quite an exciting time where there are some fantastic biotech tools, which you've already heard about this morning, that we can use to weaponize ourselves a lot better in the defense against DNA repair damage. This is a time of immense opportunity. Now, obviously, we're talking about solar radiation and galactic cosmic rays. Those are our two enemies in this. There is a reason why we talk about astronauts living in caves so much. These threats are very, very real. Here on Earth, where we have the protection of Earth's atmosphere, it's a problem enough. People do get cancer from this DNA repair damage. So there's no reason to think that when they're in space, it won't be 10 times worse, at least. And unfortunately, both galactic cosmic rays and solar radiation cause a complete double-strand break in a problem that is compounded far worse than what we see on Earth. And it's not just that we see a complete double-strand break, it's a cascading effect of other kinds of problems, chromosomal aberrations and free radicals, all those sorts of things. I'm a cancer researcher. This is deadly on Earth, so it's certainly a problem in space. Now, many of you will be aware, I know I'm preaching to the right choir here, that the Apollo astronauts actually got very, very lucky. There were some significant solar storms in and around the Apollo missions. If any of these had occurred, in some cases even just a week later, and the astronauts had been on the moon's surface at the time, there's a very strong chance they would have died. So we do have a data problem here. Actually, the overwhelming majority of our data, I borrowed this from Dr. Zubrin, I'm sure he won't mind, the overwhelming data that we have on astronauts who've been in space for long duration flights, 6 months to a year, and even longer than that, is all contained within low Earth orbit. All this data is also very recent, and it's a very small sample size. When it comes to outside of the complete protection of Earth's magnetic field, beyond low Earth orbit, we're talking a few cases of less than 2 weeks. And that is all the data that we have on this. It is quite interesting at the moment. The astronaut limit is 600 megasieverts. And we have rebels like Peggy Whitson who have gone, well, screw that. If NASA say that I've reached my radiation max, I'm just going to go work for Axiom and keep doing it because they won't say no. So we're in this sort of new world of astronauts leaving NASA to go work for Axiom because they've passed their radiation lifetime limit and are just rebelling against that and working in the private sector. So maybe we'll get some more data from that. Who knows? Go Peggy. Well done. Now, there is a paradox here in that every single one of these astronauts, including Russians who were on Mir for over a year and not working out particularly much, drinking vodka while they're on the Russian space station, not the picture of human health. None of these astronauts or cosmonauts have succumbed to cancer. None of them have ever even got cancer. So the paradox we have here is you should be falling prey to cancer, given the amount of time you're spending in space and the amount of abuse your DNA is getting. And yet everyone who has passed the year mark has not, which is odd. It's interesting why. I don't have an answer to that. But how can we make this a reality without our astronauts succumbing to cancer? Like I said, the data is very, very small. All of the recent research on this comes to the same conclusion. We just need a lot more data. But I'm going to walk you through some of the problems here. Here's my roadmap. We're going to talk about the double-strand break pathways, and I'll showcase some of our work at Reading. We're going to talk about gene editing, radiophiles, and we'll finish with the question, what happens if they get cancer while they're on Mars or on their way back? So, first of all, I have a long, long list of problems, and I hope you'll bear with me while I go through this. Of the two pathways of double-strand break repair, homologous recombination is extremely faithful, extremely accurate, very well coordinated. We're talking here about some of the most ancient proteins in the living world, proteins which we have extremely strong homologs with bacteria across the animal kingdom and the bacterial world. The Mut-S protein family, which I work on, is so similar and strongly conserved. These are ancient, very highly conserved proteins. Homologous recombination is extremely faithful and accurate. It's where there's a break, a complete double-strand break. You have a group of proteins that will find a sister strand, and they will faithfully copy the exact segment that has been broken back in, and then it will be sealed. But you also have non-homologous end joining, which is basically the DNA goes into a panic, forgive me for anthropomorphizing, and just quickly glues back on the ends so that at least it's stuck together. And you have a chunk that's missing, a chunk that's been deleted, a chunk that's been erased, but at least your strand is still intact. Now, we'd prefer homologous recombination. Unfortunately, when we're in space, our bodies tend to defer overwhelmingly to non-homologous end joining, and we don't know why. Something about being in space makes our bodies want to repair double-strand breaks in the worst way possible, and we don't understand why. There must be something that's going on biologically that is making our bodies choose the less ideal pathway. So that's problem number one. Like I've said, the majority of the focus is currently on shielding rather than the biological side. Relatively few genetics and molecular biology experiments are sent to the International Space Station. This is a bugbear of mine. Most of the genetics experiments sent to the ISS are actually by kids and students. There are competitions like Genes in Space, where children, come up with an idea and it's sent to the ISS, and it's a wonderful thing for the students. And let's see how these strands of yeast, E. coli or yeast, how does this work? And it's great, but we could really do with some really well-driven, well-funded research in genetics. There is a lot of cancer research that goes on. with cancer cells, but more needs to be done in this area. Actually, when I was doing my master's degree at Birkbeck, there was quite a dispute. I'm sure you're all familiar that protein crystals grow so much better in microgravity. I mean, going back to the days of the shuttle, a lot of protein crystal growth experiments were regularly flown to the ISS. And I asked the molecular biology department, do we do that? What do you think about that? And there's great division, great debate on this. There's no doubt that protein crystal growth and molecular biology experiments like that are much purer and higher quality when done in microgravity. It's just the cost. Am I prepared to spend so much of my budget to do that when I could get something that's not as good, but good enough on Earth? And the majority of the people at Birkbeck were like, don't send them to space, don't bother. Which is a shame, is a loss. We could learn a lot about drug discovery in space if we grew our drugs there, for example. Many of the experiments that go up are not really applicable to this, and we've got the division problem. Okay, now the data, unfortunately, has gotten worse in recent years. So I just want to bring you up to date on the last 5, 6, 7 years of DNA repair in space problems. Firstly, microgravity itself, we now know, is not friendly to DNA repair. Forget radiation, microgravity itself is problematic here. It causes an increase in apoptosis, cytoskeletal alterations, cell growth inhibition, increased mutation frequency, increased chromosome aberrations. What's more, it delays the rate of double-strand break rejoining. So not only do our bodies choose the less faithful repair mechanism, they're slower in doing it. Which is problematic, and it compounds genotoxic effects. Studies on astronauts that have spent 6 months and a year plus on the ISS showed that their telomere length is shortened. And it's shortened a lot more quickly. And when they return to Earth, it was shorter overall after returning. These correlate strongly with what we see in the negative effects of aging as well as cancer. There are higher signatures of persistent DNA damage response, including mitochondrial and oxidative stress, inflammation, and chromosomal aberrations, like I've mentioned before. So that was in a study a few years ago. Results appear to be a lot worse for women and older astronauts for reasons that we don't know why. Which is going to be problematic. If your best astronauts for your mission are women or more experienced astronauts, we need to uncover the reasons for this and see what we can do. At the moment, because of the problems that microgravity presents for DNA repair, it's hard to distinguish what are the problems that are caused by microgravity, what are the problems purely caused by radiation, and how are these informing each other. So that's another question we need answers. We can put a lot of the damage down when it comes to radiation here to linear energy transfer. Both linear energy transfer and dosage very heavily affect the DNA repair responses. We know that from a study from 3 years ago. The level and type of damage determine the cell fate more often than not, including necrosis, apoptosis, senescence, or autophagy. So double-strand breaks are the most lethal lesions that are induced by ionizing radiation, and they trigger a series of DNA damage responses. Now, personally, as a cancer researcher, I've got to add that this kind of hyperactivity causes more problems when they return to Earth as well, because it can confer tumor radioresistance. So if they get cancer, if you need to treat them through radiotherapy, you can find actually they've conferred a great deal of resistance because of this, again, which limits your options. The good news here is that 10 years ago, the Nobel Prize was awarded for elucidating the methods of DNA repair, and that's led to a transformation of cancer treatment through DNA repair therapy. We don't just cut it, burn it, and blast it anymore. If you get cancer, immunotherapy and DNA repair therapy are your best bet. They're far more successful, far more accurate, and far less invasive. Hands up if you're familiar with PARP inhibitors. Great breakthrough for breast cancer. No one. Well, look it up. It's amazing. It's absolutely amazing. So there is some positive here. I don't want it all to be doom and gloom when it comes to life on Earth. Focusing on galactic cosmic ray damage for a moment, using the DNA damage response protein 53BP1, which is a protein I work with as a biomarker, it's been found that unrepaired linear energy transfer tracks over just a 48-hour time period increased with linear energy transfer. So basically, galactic cosmic rays make the DNA repair process much more difficult. in itself. Persistent radiation-induced foci levels also make it very difficult for your B and T cells to survive. We know that your immune system suffers when you're in space. This is compounding the problem on the protein level. So there, good news. I do have good news to come. But here are the two pathways. Credit here to Nobel laureate Paul Modrich. We have homologous recombination, good. Non-homologous end joining, it does the job, but not so good. I'm just going to call it bad for now. And in space, our body wants to go that way, which is bad news. Right. It was found. This was only discovered 4 years ago, by the way, that being in space conditions impacts the choice of DNA repair pathway, leaning very heavily towards non-homologous end joining. High linear energy transfer means a high density energy along a particle track, which means a higher frequency of chromosomal aberrations, including things like translocations. Really bad news for the faithful copying of your DNA. And even deletions of sections up to 1,000 base pairs have been found by this. I mean, that's a big deal. If your body is then sticking together the break through non-homologous end joining at a loss of 1,000 base pairs, you've got problems there. So this is a problem. We know these things cause cancer in much greater frequency on Earth, and that it makes things worse for the body in space. So there's no reason to think that this isn't a problem, and there's no reason to lean on just shielding being the only solution. Let's get to some better things. This is beautiful. This is from the team I'm a part of in Reading. This is Human Mute S Alpha. Our team, the team I'm a part of, we generate accurate models. We do homology modeling of the various DNA repair proteins. Just last year, by the way, a team at NASA developed a new damage complexity score to try and help us better understand this problem. So this is something that is actively being worked on. And 2 years ago, 2023, a galactic cosmic ray damage calculation was likewise made for low Earth orbit to help us understand this. So this is something that is actively being worked on by NASA and partners. Right, so now that you're suitably scared, let me introduce you to some of our modeling. This is the homologous recombination protein ATM, nothing to do with a cash machine or anything like that. These are proteins that are actively mentioned in studies that have been done by NASA in the last 2 years. This is ATR, a partner of ATM, these complex together. And this is SPIDER, generated by using multifold 3, which is what our team does. SPIDER is one of the important proteins here for fixing your double-strand break accurately. If we can find a mechanism on Earth, the reason why I'm showing you these is if we can find a mechanism that triggers the body into choosing homologous recombination over non-homologous end joining, that would be extremely valuable. That's my top proposal for this problem. If we can find the molecular switch that gears the body towards choosing homologous recombination over non-homologous end joining, that would be incredibly helpful. Now, when it comes to non-homologous end joining, I'm talking multiple proteins are involved here. And unfortunately, it's a bit like Jenga. I don't know if you have Jenga in America. Like, you pull the lower brick out. Well, the lower bricks will, if you get it wrong, that's going to make the whole thing break down. So much like Mute S Alpha with mismatch repair, if you have a serious malfunctioning problem in one of these proteins, for example, with non-homologous end joining, it will cause the rest of it to cascade and fail. Because these proteins, they stick together like Lego bricks. They complex, and then they complex with another partner. They fix the break, and then a final pair of proteins comes and seals it up, glues it back together. They're beautiful, by the way. This is why I love biology. Biology is beautiful. Right. I'm running out of time, and I want to make sure you've got time for questions. I just included this protein because I thought it was cool that it was called Artemis. Artemis is one of the most important proteins in... Sorry, you have your hand up. Yeah.
Well, proteins in zero G, do we know that they have the same shape or?
Yes, they do. Yeah. And I'll explain why. I'll explain why now. This is Artemis bound to DNA. Let me skip ahead. So let's work on some solutions here. I wrote this article and it caused a bit of a kickback. I ended up in a debate on the radio about whether we should gene edit humans so they could live on Mars. I wish I'd known about our speakers this morning, because I should have just pointed them that way and said, oh, they'll handle it. They'll do well. For my first PhD, one of my supervisors was Lord Martin Rees. And Lord Martin Rees has written a lot on this. He's more in favour of robots going to space than astronauts. He had a debate with Dr. Zubrin at the British Interplanetary Society in London 2 years ago, which you should definitely watch. But Martin Rees is very much in favour of using gene editing on astronauts so they can suffer less on Mars. That's a much better way of doing it. This is CRISPR-Cas9. I'm sure you're very familiar with it. The Nobel Prize was awarded for it in 2020. We live in a CRISPR world now. We can do things, we have weapons, we have tools we can apply to this that we didn't have before. And it actually gets better. CRISPR-Cas9 works by inducing a double-strand break and then hijacking homologous recombination to edit the gene in the way that you want. So it relies on this. But you can now edit DNA without inducing a double-strand break and doing this through base editing. Base editing, which I think was discovered in 2021, and then prime editing as well, allow you to make precise changes to individual DNA nucleotides without inducing a double-strand break. It's like just changing one. Now, there are times when that's going to be a pain in the butt and you're going to want to just change a section at a time. But now you can change individual genes, which has been great news. I'm sure you've seen the news about Huntington's disease. These monogenic genetic diseases are going to go the way of smallpox and other diseases that are going to be ancient history because of this. Kate Rubins, amazing NASA astronaut who retired earlier this year. In 2016, she did the first ever gene sequencing in space. And then in 2018, she went one better. She did the first CRISPR in space. So we know we can do this. We know this is technology we can use while we're in space. She used a handheld device called the Minion, developed by Oxford Nanopore Technologies, just down the road from where I live, which is basically like an old school flip phone. And you can take like a piece of hair or skin cell, you can put it in, close it, and it will read out the entire genome for you in a matter of hours. The cost is about $500. Now, how can we apply this to DNA repair? In 2021, Christina Koch of the Artemis II team, she used CRISPR-Cas9 for an experiment on yeast cells on the ISS to observe their DNA repair systems. So this has been done on eukaryotes. We have actively observed, edited, and repaired DNA repair systems in space using CRISPR. So this is not too far away. In terms of radiophiles, now this was already covered this morning. On the plane on my way over here, my brother had lent me the book Project Hail Mary, and I read most of it on the plane. But I love that, without spoilers, using radiation-loving organisms to confer resistance and protection. Well, this is something that is actively talked about. You've all heard of tardigrades, I'm sure, and Deinococcus radiodurans have been mentioned many times already this morning. But we can borrow from radiophiles. We can borrow from radiophiles, we know, to help our plants be more radiation resistant. And what about us? But I'll just rush to the end now. The final thing to say on this is it is probably inevitable that astronauts are going to get cancer. And this problem is compounded by the fact that surgery in space is not really something we know we can do a lot about. But like I said, we have new weapons. We have DNA repair therapy and we have immunotherapy. And I think immunotherapy is going to be our newest best weapon for this. The Nobel Prize was awarded for this 7 years ago. Immunotherapy is non-invasive. It can be administered through injections or by oral tablets. But we are in an era of personalized precision medicine here, and space is going to make a huge amount of the difference. Because we now know if you have a tumor in space, if your DNA repair systems are breaking down and you get cancer on Mars, we can sequence your genome quickly using a handheld device. We can take a biopsy of the tumor and we can sequence the tumor signature and get that as well. Now we can use CRISPR-Cas9 to edit immunotherapy drugs and give you immunotherapy drugs in space that are tailored to the tumor that you have. And like heat-seeking missiles or like sniffer dogs, like go get that. They will go throughout your body and they will eliminate every single one of those cancer cells with that genetic signature. And then the cancer won't come back because your body is weaponized against it. That, I think, is the best final shot we've got here when it comes to a DNA repair breakdown in space. And that's all I've got time for. With that, I will happily take your questions. Thank you. You first.
Yeah, so I kind of have two questions. So it's okay.
Sure.
The first one was I understand there's like 4 end states for a cell that gets hit by radiation. It either like doesn't get damaged, it kind of just gets crippled, but it survives and lives on, sort of somehow contributing maybe to reduced degree to health of the body. It turns into like an evil cancer cell that kills you, or it just dies.
Yeah.
Is that the actual 4 states, and is it possible that you should push towards dying instead of like keeping it alive?
Yes. So DNA repair therapy, putting it in American football terms, you can go on offense or defense. You can use gene editing maybe to help fix the problem. That would be your defense. Or like with PARP inhibitors, which I mentioned earlier, you hijack the broken cells' DNA repair systems and fail them. So the protein called ubiquitin that goes around basically with Post-it notes and marks cells for destruction goes, oh, you need to die, and tags you and does that. So that's how DNA repair therapy works. So that's the first bit. I didn't have time to mention it as well, but the main cell cycle protein that's very, very important for cancer is called P53. And we have found ways of, I'm not very familiar with them myself, I just know they've been done, of upregulating P53 in people. And if you can do that for astronauts, that's a good candidate to help there as well. Making sure that the proteins that need to die, and the proteins that need fixing get tagged to be fixed. So that would be very helpful as well. Sure. Second question.
Is there I've heard that a little bit of shielding is worse than no shielding at all.
Yeah.
Is there any up-to-date information on that sort of curve between like add the shielding and get worse and then get better?
Yeah, this is something that's pretty well understood. A lot of work's done on it. If you get the shielding wrong, you can actually end up with multiple problems, as in that the bad radiation hits you, causes damage, and then it rebounds off your shielding and causes a lot of secondary damage, which is really bad. But I think that's pretty well understood. People are looking at everything from water to anything that's a high hydrogen material to do that. But I'm not a materials scientist. I'm sort of focusing on the biology side.
Any new reports or something?
Yeah, not that I have. You're at the right place.
Thank you.
Not all the cancers are, treatable with
with immunotherapy.
yes.
Yeah.
So, I mean, in fact, they're like only 30%.
Yes. Yeah.
So,
I'm going to be an optimist here because immunotherapy is so new. I mean, if you go back 10 years ago to when I was starting it as a genetics student, I was told that immunotherapy was a myth and a fairy tale. And if you want your career to go nowhere, get into immunotherapy. And then 3 years later, the Nobel Prize was awarded for it. So immunotherapy is in so many ways brand new and has already found 30% of cancers we can fix with it. We're going to get more biotechs involved, more funding is going to get into it, more universities will start teaching it, more PhD research students will take it on, more government funding will go into it. I think we're sort of at the beginning of the immunotherapy journey. There might be some that you can't do anything for because your DNA repair proteins are so ubiquitous. I think DNA repair therapy will make up the difference. But I think in 10 years, we'll be approaching a time where chemotherapy and radiotherapy could be a thing of the past. They'll be on their way out. So, and that's good news because obviously these are non-invasive.
thanks.
Thank you. Thank you very much. More questions just asked. Okay. Thank you so much for watching. Don't forget to like and subscribe. Stay tuned for more amazing conversations with incredible researchers who are changing the world we live in and driving humanity forward. Feel free to check out my Instagram at polymath_Sam or go to my website at www.sam-mckee.co.uk.